In this screencast, we're going to review a little bit about glycolysis, but then we're going to continue on with stages 2 and stages 3 of cellular respiration, including the oxidation of the pyruvate that was the product of glycolysis, and how each pyruvate continues on into the Krebs cycle. So as a review, you can take and pause this page here, but remember that this isn't such the important part over here, but the overview of the reactions of glycolysis. That first ATP is used like a match to light the fire. There's some activation energy that needs to be used, and that's enough to destabilize glucose to be split into two, and those two carbon molecules are what we're going to continue with. So in cell respiration, we can't stop at pyruvate. Not enough energy has been produced. Only a couple of ATP have been produced, and pyruvate still has a lot of energy in it, the three carbon compound that it is. There's still plenty of energy stored in those bonds. So what we need to do is to oxidize the pyruvate further, and if oxygen is available, we will enter the mitochondrion for what's called the Krebs cycle, and that way we can oxidate completely into carbon dioxide. So remember that there's some energy that is kicked out directly from glycolysis, and we're going to enter, if there's oxygen available, the pyruvate is going to enter what's called the Krebs cycle or citric acid cycle, and eventually into electron transport. But remember, so far in the cytosol of the cell, that we've also created some electron carriers, this NADH so far, and remember, we're going to harness that energy out of that later. More money in the bank. So a little review of the mitochondria structure. The big part is that it's a double membrane. Remember, it's got a folded inner membrane, and that inner membrane, the folds are called the cristae, and there's a space in between. Now, in the matrix is the space within the inner membrane, and then we've got the inner membrane itself, where a lot of the reactions are actually going to take place eventually. So it's important to know about these structures and be able to identify these, I guess, vocabulary terms as a part of the mitochondria structure. And a good question to ask here is, what types of cells would you expect? How about in the human body? Would you expect to have a lot of mitochondria? Well, the types of cells that would need to produce a lot of energy. So especially things like muscle cells or nerve cells in the body. They have a lot of mitochondria, so there's a lot of access to energy-making processes. So furthermore, almost all eukaryotic cells have mitochondria. There might be one very large one or hundreds or thousands of individual mitochondria. And the number of mitochondria, of course, like we just said, are correlated to aerobic activity. The more activity, the more energy that's needed, therefore the more mitochondria that are needed in these cells. So again, active cells would have a lot of mitochondria. We also want to visit the idea of the endosymbiosis that the mitochondria support. They divide exactly like bacteria are going to divide, and they show a lot of properties of prokaryotes, really. They actually even have their own ribosomes. They have their own DNA. They have their own protein synthesis within those organelles. And so it's hypothesized that the bacteria or other prokaryotes at one time began an endosymbiotic relationship with other cells and actually became a part of their core body makeup or their core organismic makeup, and that's the evolution of eukaryotic cells. So the second step is the oxidation of pyruvate. So pyruvate is going to enter the mitochondrial matrix. It's going to come into the mitochondrion itself. And also remember that this is happening twice. Why? Because there were two 3-carbon molecules that were the product of glycolysis. So they're going to be oxidized along the way. NAD is going to be reduced to NADH to be used later, and the product is going to be acetyl-CoA, acetyl coenzyme A, or acetyl coenzyme A, either way that you want to pronounce it. But this oxidation process is going to leave us with a product 1, a 2-carbon compound, and some carbon dioxide. And of course, we know we're going to eventually exhale that. So acetyl-CoA or acetyl-CoA is going to enter the Krebs cycle from here. So another look at it, another way to review it. You can stop the screencast here and review what you're looking at. But remember that we're going to go from a 3-carbon compound of pyruvate to a 2-carbon compound. We get rid of carbon dioxide. Remember, it's all about making ATP, right, in the end. And because CO2 is completely oxidized, it's released all the energy that it can release. It's no longer valuable. Cells get rid of it. So this third step, as we talk about it, the third step is the Krebs cycle, also known as the citric acid cycle. Well, Hans Krebs here, he recognized this or discovered it, or is credited with discovering this Krebs cycle, a huge complex pathway where each step, again, is catalyzed by a specific enzyme, as it is in every step of cellular respiration. And there is a catabolism of the 6-carbon citrate. And then again, it goes around essentially in a cycle or a cyclic manner. So the point that it evolved later than glycolysis. Remember, glycolysis the enzymes of glycolysis, the compounds involved, are very similar among all organisms. I mean, besides glycolysis and protein synthesis and DNA replication, those are processes that all life or organisms share. So the genes that code for those enzymes they're highly conserved from organism to organism throughout evolutionary change over time. They're a good measure for evolutionary studies. So we can compare eukaryotes, bacteria, and archaea, other domains of bacteria, using glycolysis enzymes. So at about 3.5 billion years ago, we can look at glycolysis as being in the cytosol, but it doesn't require a membrane-bound organelle, which is good for all organisms. That's okay. About 3 billion years ago or so, we've got evidence of atmospheric oxygen. So photosynthetic bacteria or even blue-green algae were evolved. And eukaryotes were only about 1.5 billion years ago or so, where aerobic respiration, we have evidence of aerobic respiration. Why? Because there are organelles, including the mitochondria. So of this Krebs cycle, as we go on, you can take a look at what happens to the carbons, right? So we can fulfill the universal laws of thermodynamics, that we're going to conserve the number of carbons. So matter on both sides of a chemical reaction. So start with a 3-carbon sugar, or a 3-carbon compound, the pyruvate, and it gets oxidized to acetyl-CoA, and then it enters this carbon pathway here. They're going to be the oxidation of these sugars, and along the way we're going to, of course, kick out some carbon dioxide that we'll eventually breathe out. And remember that this happens twice for every glucose molecule. So a 2-carbon sugar goes into the Krebs cycle, and it's taken apart completely, all the way down to carbon dioxide that we can kick out or breathe out. Glucose is fully oxidized. But where's the ATP? So every time the carbons are oxidized, an NAD plus is also being reduced. Remember, that will be money in the bank. What we notice here is that we're also going to reduce another electron carrier. So FADH, should have a little 2 there, FADH or NADH, it really doesn't matter. They're both electron carriers. We're going to harvest that savings a little later. We're fully oxidizing a molecule of glucose all the way to carbon dioxide in the Krebs cycle and end up with 4 ATP. Remember, we kick out some ATP once for each cycle. Remember, this happens actually 2 times per glucose. So we kick out some ATP, Remember, we've already kicked out some ATP glycolysis. So these electron carriers, remember, that they're actually hydrogen carriers. They're carrying hydrogen atoms, and what's going to happen is these electron carriers, regardless of the type, these are just two chemically different molecules that are each going to act on their own specific substrate later on, but the electron transport chain. And there we're going to harvest a lot of energy from these electron carriers. So let's review our energy accounting so far. Remember that this happens twice, what we're talking about now, because there were two pyruvate molecules. We had a couple of reductions to these electron carriers, right? There was one of these when we oxidized the pyruvate to acetyl-CoA, and then there were 3 of these in the Krebs cycle that were kicked out for a total of the 4, and there was also a reduction of FAD to FADH2, so there's another one. We also, and remember that this happens twice again, so you can double these numbers. We kicked out an ATP directly by substrate level phosphorylation here in the Krebs cycle. So here's our net gain so far just from the Krebs cycle. So we're repeating here again, but continuing to review. So stop the screencast, read through this. Remember the value of these electron carriers. We're going to cash them in soon, because this is the point, making ATP. So review, how many ATPs have we produced so far, and how much money have we put in the bank?